Welcome back to the deep dive. Today we're tackling something really fundamental, how our immune system first spots trouble.
Yeah, the very first line of defense.
Exactly. We're talking detection of pathogens, that critical initial step. Our goal here is to, cut through the jargon, make it really clear how our bodies recognize invaders, even our own damaged cells. We want it to stick, especially if you're, say, a first-year med student wading through all this info.
Think of it as building a solid foundation, because this stuff, it comes up everywhere in medicine. And we'll try to make it memorable. We've got some metaphors, some anchors, maybe a couple of acronyms to help things click. Plus, we'll throw in a few questions to get you thinking a bit deeper.
Sounds good. So we'll trace the journey, right, from that first uh-oh moment through the immediate reaction and see how that initial alert really sets everything else in motion. So let's dive in. The immune system's early warning system. Pathogen recognition. Okay, so first things first. Your immune system is basically always on patrol, right?
Constantly. Think of your leukocytes, your white blood cells, as these highly vigilant police officers.
Okay, police officers. I like that.
Yeah, they're circulating everywhere. Blood, tissues, just keep an eye out for anything suspicious. And they know what to look for because invaders, bacteria, viruses, they often wear specific uniforms, things that healthy human cells just don't have.
Uniforms?
Well, we call them PAMPs. Stands for Pathogen Associated Molecular Patterns. They're like these signature features that scream foreign invader.
Gotcha. So like what kind of features?
Oh, classic examples are things like peptidoglycan that's in many bacterial cell walls, or LPS, lipopolysaccharide from gram-negative bacteria. That one's a huge red flag for the immune system.
LPS. Yeah.
Then you've got things like lopoteichoic acid from gram-positives, mycolic acid in mycobacteria, even certain types of nucleic acids from viruses. They're all giveaways.
Okay, so that covers the external threats. But you mentioned our own cells, too.
Yeah, absolutely. It's not just about outsiders. If one of your own cells gets badly damaged, maybe from injury, lack of oxygen, infection, it starts releasing distress signals.
Like a cellular SOS.
Exactly. We call these DAMPs, damage-associated molecular patterns. Think of it as debris scattered after an explosion or smoke signals rising from injured tissue. It tells the immune system, hey, we have a problem here.
So PAMPs for pathogens, DAMPs for damage. How do those cellular police officers actually see these signals?
Good question. They have specialized receptors on their surface. Think of them as their eyes, or maybe sensors. These are the PRRs, Pattern Recognition Receptors.
PRRs, okay.
And these PRRs are specifically designed to bind to those PAMPs and DAMPs. When that binding happens, it's like flipping a switch. Alarm bells start ringing.
And are there different kinds of these PRRs?
Oh, yeah, quite a few families.
Yeah.
But two really key ones you'll hear about constantly are the toll-like receptors, or TLRs, and NOD-like receptors, or NLRs. They're major players in detecting these danger signals.
And NLRs. Got it.
And some pairings are really well defined. Like we mentioned, LPS from gram-negative bacteria, that's primarily recognized by TLR4. Viral single-stranded RNA often triggers TLR7 and TLR8. Bacterial flagellum, the little propeller, tails and bacteria use. That hits TLR5.
Wow, it's pretty specific.
It is. And DAMPs, like certain heat shock proteins released from stress cells, can be recognized by TLR4 and TLR2. Peptidoglycan fragments inside the cell, those are picked up by the NOD-like receptors.
Okay, so the PRRs on our immune cells detect a PAMP or a DAMP. The initial contact is made. But what happens next? How does that single detection event actually launch a bigger coordinated defense? That feels like a jump.
It is a jump, and a crucial one. Hold that thought, because how the system scales up from local detection to a full-blown response is really where the magic happens. We'll circle back to that too. The acute phase response, rapid reinforcements.
All right, so detection happens. The alarm is sounded locally, but the body needs to react more broadly, right? Quickly.
Exactly. That's where the acute phase response comes in. Think of it as the body hitting a general alarm button. It's a rapid systemic reaction, not just local. It mobilizes resources very quickly.
Systemic meaning throughout the whole body.
Pretty much, yeah. It's coordinated mainly from the liver, actually. Pro-inflammatory cytokines, especially one called IL-6, signal the liver.
IL-6, okay.
When the liver gets that IL-6 signal, often due to infection or injury somewhere else, it goes into overdrive, producing a bunch of special proteins, the acute phase proteins, and dumping them into the bloodstream.
And these proteins are the reinforcements?
In a way, yes.
Yeah.
Two really important ones to know are CRP, C-reactive protein, and MBL, Mannose-binding lectin.
CRP and MBL.
CRP is fantastic. It binds to a specific molecule, phosphocholine, found on many pathogens. You could anchor this. CRP is like an early warning radar, quickly detecting trouble.
Okay, CR-Bay radar. I like that. And MBL.
MBL binds to mannose, a type of sugar that's common on the surface of bacteria, fungi, viruses, but less common on our own cells, so it's another way to spot the non-self stuff.
And this CRP, is that something doctors actually measure?
Oh, absolutely. Measuring CRP levels in a patient's blood is incredibly common. If CRP is high, it's a strong indicator of inflammation or infection somewhere in the body. It's a really valuable clinical marker.
Got it. So the liver pumps out CRP and MBL as part of this rapid response.
Marking for destruction.
Okay, so we've got detection, the general alarm via the acute phase response. Now what? We need to actually deal with the invaders.
Right. And one key strategy is making them easier to eliminate. That's opsonization.
Opsonization. Sounds technical.
This means tagging or marking a pathogen. Imagine you want your phagocytes, the eating cells of the immune system, to find and destroy bacteria more easily. Opsonization is like putting a bright "eat me" sign on the bacteria.
Okay, an eat me sign. That works.
So an opsonin is any molecule that does this tagging. It enhances phagocytosis.
How does it work? Like physically?
Think of the opsonin molecule like double-sided tape. One side sticks firmly to the surface of the pathogen, the antigen. The other side has a structure that receptors on the phagocyte can easily grab onto. It creates a bridge, making it much easier for the phagocyte to latch on and engulf the pathogen.
Okay, so it connects the pathogen to the phagocyte.
Precisely. And we've already met one good opsonin, MBL. When it binds mannose on a pathogen, it can act as an opsonin.
Right, MBL.
And another really, really important opsonin is a molecule called C3b. And C3b is part of a much bigger system, IV, the complement system, a cascade of elimination.
C3b, that sounds familiar. Is that related to the complement system?
Exactly, the complement system. This is a huge part of our innate immunity. It's ancient, powerful, and works like a cascade.
The cascade, like dominoes.
That's a perfect analogy. It's a set of over 20 proteins, mostly just floating around your blood, inactive. But when the first domino is tipped over, usually by detecting microbial components, it triggers the next, which triggers the next, and so on. It's an enzyme cascade that rapidly amplifies the signal.
Okay, so this is a big amplification step. And wait, remember that question I asked earlier?
About how initial detection leads to a bigger response.
Yeah. Is this part of the answer? Does activating complement connect back to those first PAMP-PRR interactions?
Absolutely. That initial detection, the release of inflammatory cytokines, that's often what tips over the first domino in the complement cascade. Or sometimes, complement proteins can directly recognize pathogen surfaces themselves. It's a major way the body escalates from threat detected to full-scale defense activated.
Okay, wow. So complement is a key amplifier. How does it get started? Are there different ways?
Yes, there are three main activation pathways, but they all kind of converge towards the same powerful outcomes.
3 pathways, okay.
First, the classical pathway. Think classical because it often involves antibodies, which are part of the adaptive immune response we'll touch on later. When antibodies bind to a pathogen, they can recruit the first complement protein, C1, and kick things off. It involves C1, C2, C4, ultimately forming something called C3 convertase. Anchor. Classical needs antibodies.
Okay, classical antibodies. What else?
Then there's the alternative pathway. This one is really cool because it can be triggered directly by bacterial or fungal cell wall components. No antibodies needed. It involves different factors, like B and D, interacting with C3 itself, setting up an amplification loop. Anchor. Alternative is direct detection.
Direct detection. Nice. And the third?
The lectin pathway. This one starts when our friend MBL, the Mannose-binding lectin.
MBL again.
Yeah. When MBL binds to mannose on a pathogen, it recruits other factors that activate C4 and C2, similar to the classical pathway, also leading to that C3 convertase. Anchor. Lectin pathway for MBL.
Okay, so classical antibodies, alternative direct, lectin, MBL. But they all lead to this C3 convertase thing.
Exactly. C3 convertase is like the central hub. Its job is to chop up lots and lots of C3 protein. And when C3 gets chopped, it generates that C3b opsonin we mentioned.
The eat me tag.
Right. And it also generates other fragments. From this central point, complement unleashes 3 main effects.
3 big outcomes. What are they?
1. Inflammation. Certain fragments like C5a, C4a, and C3a are powerful inflammatory signals. They recruit more immune cells to the site.
Okay, inflammation boost. 2.
2. Opsonization. We already mentioned C3b coating the pathogen. Other complement components like C4b and even C1q can also act as opsonins, making pathogens even easier to phagocytose.
More eat me signs. Got it. And 3.
3. Direct lysis. Killing the pathogen directly. This happens via the membrane attack complex, or MAC.
Amazing.
Yeah, it's formed by a sequence of later complement proteins C5b through C9. They assemble together and literally punch holes, like pores, right through the pathogen cell membrane. Water rushes in, the cell bursts. It's pretty dramatic. Acronym, think MAC attack.
Wow. Inflammation, opsonization, and direct killing via MAC attack. Complement is seriously powerful.
It really is a crucial rapid response weapon via the acute inflammatory response containing the fire.
So we have detection, amplification via complement, tagging via opsonins,
Mm.
all leading to this localized battle, right? The inflammation we actually see.
Precisely. That's the acute inflammatory response. It's the body's immediate, localized strategy to contain the threat. Usually kicks in within hours, well before the more specialized adaptive immune system is fully geared up.
And it's driven by all the things we've just talked about.
Yes, innate immune cells like macrophages started off releasing those pro-inflammatory cytokines. Complement activation pours fuel on the fire, and it leads to those classic signs.
Redness, swelling, heat.
Exactly. Caused by key steps. First, vasodilation, blood vessels widen to increase blood flow to the area. Second, increased vascular permeability. The vessel walls become leaky, letting fluid, plasma proteins like complement and antibodies, and importantly, immune cells escape into the tissue where the problem is.
Okay, easier access for the troops.
Right. Third, the release of loads of inflammatory mediators, cytokines, chemokines, histamine, which orchestrate the whole process. And fourth, crucially, the migration of neutrophils.
Neutrophils. Those are the first responders.
They absolutely are. They follow the chemical trails, squeeze out of the blood vessels, and swarm the site of infection. They're phagocytes, expert eaters of bacteria and debris.
Okay, quick recall question for you listening. If neutrophils are rushing to an infected wound, what's their main job once they get there?
Phagocytosis. They're there to engulf and destroy the pathogens, eat up the bad guys.
Perfect. So, acute inflammation is about containing the fire locally, getting those neutrophils in fast.
The immune system's communication network.
Now, underpinning all of this coordination, the alarm, the recruitment, the response is communication. And the language the immune system uses is largely spoken through cytokines.
The messenger molecules.
Exactly. Tiny proteins released by cells that signal to other cells, telling them what to do, where to go, whether to ramp up or calm down. It's an incredibly complex network.
Can we highlight a few key ones, like the main players we should know?
Definitely. We mentioned IL-6, already key for that acute phase response, telling the liver to make CRP.
Right, IL-6 connects to CRP.
Got it. Then there's IL-1, often released by macrophages early on. It causes fever, which can help fight infection, and activates other immune cells.
Okay, IL-1, fever.
IL-8 is super important. It's a chemokine, meaning it creates a chemical trail. Its main job is neutrophil chemotaxis, calling those neutrophils to the site of infection.
IL-8 is the neutrophil siren.
Pretty much. IL-12, often from macrophages and dendritic cells, is key for activating natural killer cells and pushing the adaptive response towards a certain type called Th1.
Okay, IL-12 for NK cells and Th1.
TNF-alpha is another big pro-inflammatory one. Increases vascular permeability, helps activate cells, but too much can cause problems systemically.
Right, TNF-alpha, powerful, but needs control.
Then you have cytokines more associated with allergy or parasites like IL-4, IL-5, and IL-13, often linked to a Th2 type response, IgE antibodies, and eosinophils.
Allergy-related, okay.
And IFN gamma, interferon gamma, is crucial, especially for fighting viruses and intracellular bacteria. It's produced by Th1 cells and NK cells, and its main job is to superactivate macrophages, making them much better killers.
IFN gamma activates macrophages. Okay, another recall connection. If you have an infection like tuberculosis, where the bacteria live inside macrophages, which cytokine is vital for helping those macrophages fight back?
That would be IFN gamma, produced by Th1 cells to boost macrophage killing power.
Excellent. And is there anything that, calms things down?
Yes. IL-10 is a major anti-inflammatory cytokine. Often produced by regulatory T cells, its job is to suppress immune responses and prevent them from going overboard or damaging tissues. It's the, okay, stand down, sit on...
IL-10, the peacekeeper. That's a lot of messengers.
It is, but understanding the main roles of these key players really helps connect the dots in how the immune system manages different threats. 7. Antigen presentation, the bridge to adaptive immunity. So while all this innate stuff is happening, the PAMPs, PRRs, complement, inflammation, cytokines, something else crucial is starting. It's the handover.
The handover to what?
To the adaptive immune system, the part that's highly specific and develops memory. The link between the immediate, nonspecific innate response and that specific adaptive response is antigen presentation.
Ah, right. Showing the enemy's face.
Exactly. Specialized cells, mainly APCs, antigen-presenting cells, like dendritic cells and macrophages, engulf the pathogen, chop it up into little pieces, antigens, and then display those pieces on their surface.
Like putting up a wanted poster.
Perfect analogy. They present these antigens, usually nestled in special molecules called MHC proteins, to T cells. This tells the T cells precisely what the enemy looks like so the adaptive system can launch a targeted attack.
And which cells do this presenting? You mentioned dendritic cells and macrophages.
Those are the main professional APCs, yes. B cells can also present antigen. They all express a specific type of MHC molecule, MHC class II, which is crucial for presenting antigens to helper T cells. Quick recall. Which cells express MHC class II?
Macrophages, dendritic cells, and B lymphocytes are key APCs.
Exactly. This presentation step is the vital bridge, initiating that highly specific, powerful adaptive response. 8. Natural killer NK cells. Innate assassins.
Before we fully cross that bridge, though, there's one more innate player we should highlight. Natural killer cells or NK cells.
Ah, yes, the NK cells. They're fascinating. Part of the innate system, but kind of unique. Think of them as innate assassins.
Assassins. How do they work?
They have a really clever surveillance mechanism. Healthy cells in your body constantly display a molecule called MHC class I on their surface. It's like an ID badge saying, I belong here, I'm healthy.
Okay, MHC, means friendly.
Right. NK cells have inhibitory receptors that recognize MHC-I. When they see it, they're inhibited, they leave the cell alone.
But what if a cell is not okay?
Well, some viruses force infected cells to hide their MHC-I, trying to evade detection by T cells. And some cancer cells also lose MHC-I.
Ah, so they ditch their ID badge.
Exactly. And when an NK cell encounters a cell that's missing MHC-I, its inhibitory signal isn't triggered. Instead, activating signals take over, and the NK cell kills the target cell.
How does it kill it?
It releases toxic granules containing proteins like perforin and granzymes, which induce apoptosis-programmed cell death in the target cell. It eliminates cells that are trying to hide.
That's brilliant. So, another quick connection. If a virus makes infected cells downregulate MHC-I to hide from cytotoxic T cells, which other immune cell is perfectly placed to spot and kill these MHC-I deficient cells?
That's the NK cell's specialty.
NK cells, got it. The assassins of the innate world. IX. The adaptive immune system. Specialized warfare. Okay, so the innate system does all this amazing detection, amplification, and initial containment, and then hands off the specific intel via antigen presentation. This kicks off the adaptive immune system.
Right, the highly specific memory-forming branch.
Yeah.
And it generally operates along two main arms, depending on the type of threat.
Two arms.
Yeah. First, cell-mediated immunity. This is often called the Th1 response. Think of it as specializing in threats inside our cells.
Intracellular threats, like viruses.
Exactly. Viruses, some bacteria that live inside cells, some fungi. The main players here are macrophages, super activated by IFN gamma, remember, and CD8 plus T cells, also known as cytotoxic T lymphocytes, which directly kill infected cells. This whole arm is orchestrated by Th1 helper cells.
Okay, Th1 cell, mediated for intracellular stuff, kills infected cells. What's the other arm?
Humoral immunity, often called the Th2 response. This arm primarily targets pathogens outside our cells in the bloodstream in tissue fluids.
Extracellular threats, like most bacteria.
Yep, most common bacteria, some fungi, protozoa, and bigger things like parasitic worms. The main effector cells here are B cells, which mature into plasma cells that pump out antibodies. Eosinophils and mast cells also play roles, especially in parasite and allergy responses. This is orchestrated by Th2 helper cells.
So EC2 humoral for extracellular stuff using antibodies. That's a clear division.
It's a helpful distinction, yeah. And comparing the main lymphocytes involved, T cells and B cells, highlights some key differences.
Okay, T cells versus B cells. Quick rundown.
Sure. Both recognize specific antigens. Both multiply like crazy when activated. That's clonal expansion. But T cells need that antigen presented to them by an APC on an MHC molecule. They recognize processed little bits.
Right, MHC restriction for T cells.
Whereas B cells can recognize whole intact antigens, like proteins on a bacterial surface, directly through their B cell receptor, which is basically a surface-bound antibody, usually IgM initially. They don't need MHC presentation for recognition.
Ah, okay, big difference.
And B cells are the ones that actually synthesize and secrete antibodies. T cells have different jobs, helping, killing, regulating. Also, T cells mature in the thymus, B cells in the bone marrow.
T for thymus, B for bone marrow. Easy anchor. X. Clinical significance. Why this matters to you. We've covered a huge amount of ground, from PAMPs to MBSC tags to T and B cells. Why is getting a solid grip on all this so critical for someone going into medicine?
Oh, it's fundamental. Understanding immunity is key to so many areas. Diagnosing infectious diseases often relies on detecting immune responses like antibodies or inflammation markers like CRP. Managing conditions where the immune system goes wrong, immunodeficiencies where it's too weak, or autoimmune diseases where it attacks the self requires knowing how it should work.
Right, you need the blueprint to understand the malfunction.
Exactly. And think about vaccination. The entire principle is based on triggering a specific adaptive immune response and generating memory using these very mechanisms.
Yeah, absolutely.
Even cancer therapy is increasingly leveraging the immune system. Cancer immunology is a massive exploding field. So yeah, understanding immunity, it's everywhere in clinical practice. Immune responses to specific pathogens tailor defense. And what's really elegant is how the immune system tailors its response based on the type of pathogen it encounters.
It doesn't just use a one-size-fits-all approach.
Not at all. Take bacteria. Most are extracellular, right? So the body typically mounts a strong Th2 humoral response antibodies, complement activation, phagocytosis boosted by opsonization.
Makes sense. Antibodies tag, complement, helps kill or tag more.
But some bacteria are clever and hide inside cells things Neisseria, Salmonella, Chlamydia, mycobacteria like TB. For those, you absolutely need a Th1 cell-mediated response with activated macrophages and cytotoxic T cells.
Okay, so intracellular bacteria need Th1. What about viruses?
Viruses always replicate inside cells, always. So the primary defense is always a Th1 cell-mediated response. NK cells are crucial early on, then cytotoxic T cells come in to kill infected cells. IFN gamma is vital. Antibodies against viruses are made, sure, but they're mainly useful for preventing infection or reinfection, and for diagnostic tests, serology.
Got it. Viruses each 1-0.
Fungi. Fungi are usually extracellular, so often a Th2 response initially. But some, like Cryptococcus or Pneumocystis, can be intracellular, especially in immunocompromised people. Then again, you need that Th1 response.
Okay, connects to patient status, like an HIV patient with Pneumocystis pneumonia.
Exactly. In that patient, the critical defense should be Th1-mediated cellular immunity because it's acting like an intracellular fungus.
Oh, yeah.
But HIV damages the T cells needed for that response. That's why it's such a common opportunistic infection in AIDS.
Yeah, that really clicks. What about parasites, worms, and protozoa?
Right. Protozoa and helminths. Parasitic worms are obviously extracellular. They're huge. They trigger a strong Th2 response, lots of IgE antibodies, and specialized cells like eosinophils get involved. Some protozoa, though, like Plasmodium, which causes malaria, live inside cells for part of their life cycle, so they also trigger a Th1 response.
Okay, so the location intracellular versus extracellular is the key determinant for Th1 versus Th2.
It's a major driver, yes?
When the defenders are weakened.
Now, what happens when this amazing system breaks? When it's weakened? Immunodeficiencies.
Right, and they broadly fall into two camps. Primary immunodeficiencies, or PIDs, are usually genetic, inherited, often show up in childhood.
Like built-in defects.
Exactly. If the defect is mainly in B cells, you see severe recurrent infections with bacteria, the kind usually handled by antibodies.
Yeah.
If it's a T cell defect, the big problem is infections with intracellular pathogens viruses, those intracellular fungi and bacteria we mentioned.
Makes sense, based on the Th1, Th2 roles.
Yep. And SCID, severe combined immunodeficiency, affects both B and T cells. These kids get all sorts of recurrent infections, fail to thrive. It's devastating without treatment like a bone marrow transplant. There are also phagocyte disorders affecting neutrophils or macrophages, leading to bacterial and fungal infections. And complement defects increase susceptibility to certain bacteria, especially Neisseria, causing meningitis, and oddly, also increase the risk of autoimmune diseases.
Okay, those are primary, genetic. What's the other camp?
Secondary immunodeficiencies, or SIDs. These are acquired later in life due to other factors.
Acquired how?
Malnutrition is a huge one globally. Immunosuppressive drugs used for transplants or autoimmune disease. Chemotherapy. Infections themselves, most famously HIV/AIDS, which devastates helper T cells. Or even something like having your spleen removed asplenia makes you vulnerable to specific encapsulated bacteria because the spleen is important for clearing them.
So lots of ways the system can be weakened after birth. 13, autoimmune disease. When the defenders attack themselves.
And the flip side of immunodeficiency is when the immune system is too active or active in the wrong way, attacking the body's own tissues. That's autoimmune disease.
A failure of self-tolerance.
Precisely. The system loses the ability to distinguish self from non-self and mounts an attack against its own cells or proteins.
What causes that? Why does it happen?
It's complex, often a mix of genetic predisposition and environmental triggers. Infections are thought to play a role sometimes, maybe through molecular mimicry.
Molecular mimicry.
Yeah, the idea is that a pathogen has an antigen that looks very similar, structurally, to one of our own self-antigens. The immune system mounts a response against the pathogen, but then those antibodies, or T cells, mistakenly cross-react with the similar-looking self-tissue.
Ah, like a case of mistaken identity.
Exactly. Or sometimes antibodies made during an infection might just happen to bind to self-antigens. There can also be failures in the regulatory mechanisms that normally keep self-reactive T cells in check.
Can you give an example?
A classic one is acute rheumatic fever, ARF. It happens after a group A strep throat infection. The person makes antibodies against the strep bacteria, but unfortunately a protein in the bacteria, M protein, looks structurally similar to proteins in the human heart muscle, like cardiac myosin. So those anti-strep antibodies cross-react and attack the heart valves.
Wow. So the infection is gone, but the immune response damages the heart.
Exactly. It typically shows up 2 to 4 weeks after the sore throat. Causes carditis, heart inflammation, arthritis, jerky movements called chorea, skin rashes. The classic signs of rheumatic fever. It's a prime example of molecular mimicry leading to autoimmune damage. Outro.
We have really covered the waterfront today, from that very first PAMP detection through the acute phase, the complement cascade lighting things up, the specific Th1 and Th2 responses, all the way to what happens when the system is weak or attacks itself.
It really underscores how interconnected and, frankly, elegant the whole system is. Every component, from a single receptor like a TLR to a complex cascade-like complement, plays a critical role. It's constantly adapting, learning, protecting.
It truly is amazing, and understanding these foundations, it isn't just academic, right? It unlocks so much about health, disease, treatment.
Absolutely.
So here's a final thought to leave you with. We've talked about how the immune system communicates using cytokines, how we can talk to it with vaccines. But what other languages might it be speaking that we're only just starting to tune into? Think about the gut microbiome's influence, or the nervous system's connection to immunity. How might deciphering those languages revolutionize medicine in the future?
A fascinating question. There's still so much to learn.
Indeed. Well, thank you for joining us on this deep dive into the immune system's frontline. Keep exploring, keep questioning.